Following his resignation in 1982 from the Harvard faculty to take the reins of Biogen, the company promptly lost $11.6 million in 1983 and $13 million in 1984. Understandably, Gilbert took refuge behind ivy-covered walls, returning to Harvard in December 1984. But Biogen continued to lose money after his departure. It was hardly the stuff of a mouth-watering investment prospectus. But ultimately, Gilbert's grand plan foundered, owing more to circumstances beyond his control than to any managerial shortcoming. The stock market crash of October 1987 abruptly terminated Genome Corp's gestation. In fact, Gilbert was guilty of nothing as much as being ahead of his time. His plan was not so different from the one Celera Genomics would implement so successfully a full 10 years after Genome Corp was stillborn. And the concerns his venture provoked about the private ownership of DNA sequence data would come into ever sharper focus as the HGP progressed. The plan our Gilbert-less NAS committee devised under Alberts made sense at the time. And indeed, the Human Genome Project has been carried out more or less according to its prescriptions. Our cost and timing projections have also proved respectably close to the mark. Knowing, as any PC owner has learned, that over time technology gets both better and cheaper, we recommended that the lion's share of actual sequencing work be put off until the techniques reached a sensible, cost-effective level. In the meanwhile, the improvement of sequencing technologies should have high priority. In part toward this end, we recommended that the smaller genomes of simpler organisms be sequenced as well. The knowledge gained thereby would be valuable both intrinsically as a basis of enlightening comparisons with the eventual human sequence, and as a means for honing our methods before attacking the big enchilada. Of course, the obvious non-human candidates were the geneticists' old flames: E. coli, baker's yeast, C. elegans, the nematode worm, popularized for research by Sidney Brenner, and the fruit fly. Meanwhile, we should concentrate on mapping the genome as accurately as possible. Mapping would be both genetic and physical. Genetic mapping entails determining relative positions, the order of genetic landmarks along the chromosomes, just as Morgan's boys had originally done for the chromosomes of fruit flies. Physical mapping entails actually identifying the absolute positions of those genetic landmarks on the chromosome. Genetic mapping tells you that gene 2, say, lies between genes 1 and 3. Physical mapping tells you that gene 2 is 1 million base pairs from gene 1, and gene 3 is located 2 million base pairs further along the chromosome. Genetic mapping would lay out the basic structure of the genome. Physical mapping would provide the sequencers, when eventually they were let loose on the genome, with fixed positional anchors along the chromosomes. The location on a chromosome of each separate chunk of sequence could then be determined by reference to those anchors. We estimated that the entire project would take about 15 years and cost about $200 million per year. We did a lot more fancy arithmetic, but there was no getting away from Gilbert's $1 per base per pair estimate. Each space shuttle mission cost some $470 million. The Human Genome Project would cost 6 space shuttle launches. Our report was published in February 1988. The rough draft of the genome was published in 2001. The gaps continue to be filled in by sequencing labs around the world as I write. And in 2003, the 50th anniversary of the discovery of the double helix and the 15th of the committee's report, we will see the completion of the sequence. While the NAS committee was still deliberating, I went to see key members of the House and Senate subcommittees on health that oversee the NIH's budget. James Weingarten, head of NIH, was in favor of the Genome Project from the very start, as he put it. But less far-sighted individuals at NIH were opposed. In my pitch for $30 million to get NIH on the genome track, I emphasized the medical implications of knowing the genome sequence. Lawmakers, like the rest of us, have all too often lost loved ones to diseases like cancer that have genetic roots, and could appreciate how knowing the sequence of the human genome would facilitate our fight against such diseases. In the end, we got $18 million. Meanwhile, the DOE was able to secure $12 million for its own effort, mainly by playing up the project as a technological feat. This, one must remember, was the era of Japanese dominance in manufacturing technology. Detroit was in peril of being run over by Japan's automobile industry, and many feared the American edge in high tech would be the next domino to fall. Rumor had it that 3 giant Japanese conglomerates, Mitsui, Fuji, and Seiko, had combined forces to produce a machine capable of sequencing 1 million base pairs a day. It turned out to be a false alarm, but such anxieties ensured that the U.S. Genome Initiative would be pursued with the sort of fervor that put Americans on the moon before the Soviets. In May 1988, Weingarten asked me to run NIH's side of the project. When I expressed reluctance to forsake the directorship of the Cold Spring Harbor Laboratory, he was able to arrange for me to do the NIH job on a part-time basis. I couldn't say no. 18 months later, with the HGP fast becoming an irresistible force, NIH's Genome Office was upgraded to the National Center for Human Genome Research. I was appointed its first director. It was my job both to pry the cash away from Congress and to ensure that it was wisely spent. A major concern of mine was that the HGP's budget be separate from that of the rest of NIH. I thought it vitally important that the Human Genome Project not jeopardize the livelihood of non-HGP science. We had no right to succeed if by our success other scientists could legitimately charge that their research was being sacrificed on the altar of the megaproject. At the same time, I felt that we, the scientists embarking on this unprecedented enterprise, ought to signal somehow our awareness of its profundity. The Human Genome Project is much more than a vast roll call of A's, T's, G's, and C's. It is as precious a body of knowledge as humankind will ever acquire, with the potential to speak to our most basic philosophical questions about human nature for purposes of good and mischief alike. I decided that 3% of our total budget, a small proportion but a large sum nevertheless, should be dedicated to exploring the ethical, legal, and social implications of the Human Genome Project. Later, at Senator Al Gore's urging, this was increased to 5%.